Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Most cancer cells have genomes that are much less methylated than those of normal cells, but whether this loss of methylation, an epigenetic process, has any functional meaning for the cells has long been an unanswered question. Now researchers show that the loss of DNA methylation across the genome changes the timing of DNA replication and alters the shape of the 3D compartmentalization of DNA, which helps steer gene expression. The study, published September 21 in Cell Reports, is an elegant dissection of the impact of DNA methylation on 3D genome organization, says Emma Bell, a bioinformatician at the Princess Margaret Cancer Centre in Toronto who did not participate in the work. It's really important to show how extensively aberrant DNA methylation, which is a common artifact of cancer, impacts higher-order genome organization and DNA replication. For the past 25 years or so, Susan Clark and her group at Garvan Institute of Medical Research in Australia have been interested. Clark and colleagues hypothesized that DNA methylation plays a role in the maintenance of this genome architecture, and, in the new study, used colorectal cancer cells to investigate that hypothesis. The researchers started with two versions of the same cells, one normal cancer cell line and one with knockouts of two DNA methyltransferase enzymes, which copy the methylation pattern of a DNA strand to daughter DNA strands generated during replication. The cells lacking DNA methyltransferases had reduced genome-wide methylation levels. Then, in individual cells from both cell lines, they mapped DNA replication throughout the genome and investigated the genome's 3D organization. The team found that hypomethylation resulted in a shift in replication timing, that is, how early or late during replication a region was copied. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.